Hello and welcome. This is Bhaskar Napte from Pharma Growth Hub. Hope you are doing great today. So as a part of today's video, we are going to discuss about few of the very important frequently asked questions about out of specification procedure. Now these are the few questions which I often get during my live webinars. So I thought of sharing this with you as well. I hope you will get benefited out of this video. So let me zoom in a little so that you will be able to see screen clearly. Yes, and this is the first question I often get during my live webinars, and sure it is on to the screen. Do we need to raise an OS if OS result is observed during API method transfer? Right? So if OS result is observed during API methods transfer, you are transferring a method for drug substance and you come across the out of specification result. Now, this question has to be answered with respect to what is the location or at which location the OS result is observed, because you know that the method transfer activity gets happened at two different locations. The first one is at originating lab and the second one is at receiving lab. So it is very important for you to give this answer with respect to the location. So at originating lab means the lab which has validated the test procedure, has experience of dealing with the test procedure. In case if that lab has come across with the OS result for the API under method transfer, then certainly OS is required, because this is the lab which might have released the same API batch earlier. This is a lab which is well experienced in handling this test procedure. So the OS must be taken in case if the originating lab has come across the out of spec result. The moment you come across the out of spec result, and as this batch is already released in the past, you have to take care of these three important points. The first and foremost, you need to inform to API vendor, API supplier, API manufacturer about this failure. If you are not the API manufacturer, right, and if you are getting API from some another vendor, you must inform to the API vendor, because this is the lot which might have supplied to many another vendors. And in case if this particular lot gets used into the uh, production of drug product, then it has a very serious impact. So for that reason, you must also inform to the API's vendor. Investigate the release analysis data. So as this API lot was released earlier during the uh, its release analysis, it is very important for you to also look into the original initial release analysis data. And in case the same API lot has been used for manufacturing of the drug product, then you have to also assess the impact of the APIs out of specification onto the drug product's quality. So this is how you should deal with the out of spec result if occur at the originating lab. In case if the out of spec result occurs at receiving lab, now this is the lab which is experiencing handling this test procedure for very first time, and they do not have much experience of handling this particular test procedure. So the errors are quite obvious, and for that reason if it happens at the receiving lab, the OS should not be the appropriate procedure to investigate the lab errors, but you may investigate the lab error with the lab deviation or lab incident test SOP. So this is the answer you must give while the OS observed during the analytical method transfer. Always bifurcate the answer with respect to failure at originating lab and receiving lab. I hope you are clear on the first question. Let us now understand the second question. Do we need to raise an OS if OS result is observed for vendor evaluation API sample? So you are evaluating the sample received from the API supplier who is just under validation or the process of vendor qualification, and during that particular evaluation sample the OS is observed for one or multiple test procedure. So are you going to investigate that failure through OS procedure? The answer could be no. Answer could be no, because there is no much impact onto this OS. You are just validating the vendor. I mean, you are just qualifying the API vendor, right? And because of that reason, as there is no much risk in failure of the specification for this particular API lot, you may need to go through the OS procedure, but certainly you can investigate the failure through the lab incident or the lab deviation. And similarly, don't forget to communicate to the vendor about this particular observation. Okay, so let us now move on to the question number 3. Do we need to raise OS if OS result is reported whereas bracketing standard is failed? So that as a part of your running sequence, you have analyzed, let us say, multiple samples in one sequence, and you are calculating the result, and you found that, okay, now one of the result is out of the spec. For example, you are analyzing assay for paracetamol API, and there are, let us say, 7 different lots, and you loaded these 7 different lots in a same sequence, right? And the sequence is running, you are evaluating the sample, and you found that out of the 7 batches, the batch number 2 has got the out of spec assay result. Subsequently, when the sequence got completed, you have injected the bracketing standard, because you also need to confirm whether the system was suitable from its start to end. Now there was the out of spec result for batch number 2, and when you completed the bracketing standard, you found that, okay, now the bracketing standard itself is not meeting my system suitability criteria. The question to you is, are you going to investigate that batch number 2 assay failure through out of spec procedure? The response to this question should be no. The reason is because, look at the your system suitability requirement. Your system suitability requirement says that your initial standard must meet the certain percent RSD, tailing factor, or theoretical plate count or resolution, and then followed by you have injected the bracketing standard. And another criteria is what, now your bracketing standard also must meet the system suitability criteria. But now in this case, your bracketing standard is not within the system suitability requirement. You can say, now my system was not suitable during the analysis, and hence whatever result I reproduced or generated are not valid. And if the result are not valid, I must not report them at all. There is no reason why I must report the assay result for either failed batch or the passed batch, isn't it? Having said that, if there is no result reported, there is no question of raising an OS. But you must investigate the failure through your lab deviation or lab incident SOP. Though I have not mentioned over here, but please consider investigating the system suitability failure through the incident procedure. Let us go move on to the point number 4, and here it is. The question is, do we need to raise an OS if OS result is observed for swab or residue or rinse analysis? So you got the your cleaning validation sample or cleaning verification sample. It can be swab sample, it can be rinse sample, it can be any kind of sample, but it belongs to a cleaning verification or cleaning validation. The question to you is, do we need to raise an incident for the failed swab sample? The answer to this question is obviously yes, right? Uh, the reason is probably I need to correct over here. In case if the swab sample belongs to a cleaning validation, probably you have something to say that the method is not yet validated. I mean, the cleaning method is not yet validated. We really do not know whether the given cleaning procedure is going to meet the expected outcome. And for that reason, if it is out of spec, I may not investigate the failure through out of the spec, isn't it? But you can certainly raise an incident or lab deviation, and through that, you need to identify the failure in the swab result. Is it really a lab error or is it really a ineffective cleaning procedure? But in case if the swab results are observed in verification, that you are taking the another batch, and as a part of your routine process, you supposed to first understand the carryover and the swab limit has been defined. So for those situations, in case if the swab result is out of the spec, is beyond your the maximum allowable carryover, your macro value, then certainly you need to investigate that swab failure through out of the spec procedure. I hope you are clear in between cleaning validation. If the swab result gets failed, what you supposed to do? And in case if you are just verifying the swab result, what you supposed to do? Thank you so much. Let us go on to the point number 5 now. The question number 5. Do we need to raise an OOS if API result is out of the spec during working standard qualification? Okay. Now this is very interesting question. So you are, you know, preparing the working standard for your API from your API lot, and during the analysis you found that okay, now the let us say the assay of the API lot is not meeting the spec. So do you need to raise an OS? Understand, now this question also needs to be answered in two different ways. Need to understand if API is used only for working standard qualification. Sometimes we get the API from the supplier just for working standard qualification, right? So you get, let us say, 100 gram of the API, and now you are making the working standard out of that 100 gram quantity of the API. So what you are going to do? See, this particular API lot is not used for your drug product manufacturing. So there is no risk at all. You are just receiving the sample from the API vendor to qualify working standard at your lab, and the assay is out of the spec, for example, and you need not to investigate the failure through out of the specification procedure. Having said that, you again need to investigate this failure through your lab incident or lab deviation procedure, and don't forget to inform to the API vendor about this particular lot's failure. But in case the second situation, in case if the API lot is used for manufacturing of drug product, the same API batch you are now using for preparation of the working standard and found to be out of the spec, you must take the you must investigate the failure now through out of the specification procedure. So again, inform to the API vendor, investigate the release analysis for the API if it is released earlier. Of course, because you said that it has been used for the manufacturing of the drug product, then this lot must have analyzed earlier. And don't forget to assess the impact on the drug product's quality. So these are the three important points. And the last but not the least in this video is, if the OS result is found during out of the trend or incident investigation or OOG investigation, do we need to raise an OS? Now this is little tricky question. You are investigating the failure observed because of your lab incident, lab deviation, or the out of the trend or out of the expectation result. So are you going to raise an OS if the investigation results are out of the spec and the investigation is conducted as a part of your OOT or incident or the OE observation. Okay, so let us now answer this question in two different scenarios. The first one is if hypothesis is just as per test procedure. See, as a part of investigation of OOT, incident or OE, you are building some hypotheses. The hypotheses are built to understand the impact that you are thinking that okay now the analyst might have used wrong filter paper. So wrong filter paper is just your doubt that okay this might have resulted into the out of the spec result, and then you are going to bring the hypothesis around the filter paper. If I use the wrong filter paper, if I use the right filter paper, if I do not saturate the filter paper, so there could be multiple hypothesis experiment you can build. Now for some reason, if the hypothesis is just as per the standard test procedure, right, the hypothesis is just as per the standard test procedure, and if you come across the out of the spec result, now what do you think? Do you need to raise an OS? Certainly yes, because you are not deviating from as such procedure and the result is out of the spec. That may ask you to investigate the failure through the OS. But I have a question for you now. The hypothesis testing should not be as per actual test procedure. Sometimes you may tend to do that, but ideally speaking, the hypothesis testing should not be as per your actual test procedure, and that's the justification I'm just trying to give over here. And I will read for you the hypothesis. Ideally, hypothesis should not be exactly as per STP. If you allow same test procedure as a hypothesis, that indicates you are trying to check whether sample meets the specification or not. You are just trying to understand whether the sample complying to the assay if I just retest it as a part of hypothetical testing. I hope you understand the criticality. While if someone just retest the sample under some heading of the hypothetical analysis, but just following the same test procedure, it is sometimes most of the times it is highly questionable. The second scenario is quite obvious. If hypothesis testing is not as per the STP, you are challenging your STP, decreasing the sonication time, increasing the sonication time, you are making some changes in the dilution that indicates that you are not actually following the test procedure. You are just trying to understand what happens if I increase the sonication. Am I getting the higher value? What happens if I reduce the sonication time? Am I getting the lower assay value? So this is not actually as per the test procedure. And if this hypothesis analysis is not actually as per the test procedure, there is no question of investigating the failure even, and there is no question of taking the OS. I hope you are now clear in which situation you know during the investigation of any kind of failure, OOT or out of spec, or in case of lab deviations, you need not to take an OS. Let me also explain you further, and this is much better explanation. Ideally, hypothesis testing must not be exact as per the STP. Hypothesis testing is done to identify root cause. Hypothesis testing may be with certain modification in test procedure, and during hypothesis testing, root cause is not yet clear. You are not very much clear about the exact root cause, and hence reported result maybe pass, maybe fail, may be attributed to the testing error that is still hidden, and you are not able to identify that, as we are not sure about reported value whether confirmed or pass or fail. The result reported during hypothesis testing should not be considered as a valid, right? So this is the reason why during hypothesis, you know you should not raise the OS in case even though the result found out of the spec. And this is just one example of hypothetical testing. Exact method says crush the tablet into fine powder, right? And as a part of hypothesis, you say crush the tablet into coarser powder to prove non-homogeneous sample resulted into an out of the trend result. So this is just an example how you can build hypothesis. So I hope you are clear about why the hypothesis testing need not to be investigated through OS. And make sure that your hypothesis experiment should not be just like the retesting as per the actual test procedure. That looks to be some fishy. You are just trying to understand. You are just naming it as hypothesis testing, but you are actually conducting the as such test procedure, trying to understand whether I am getting the result into a compliance. So these are the six questions, and I hope you must have found these questions interesting. Thank you so much.